In this lecture, we'll look at hemoglobin and oxygen transport, as well as carbon dioxide transport. As I mentioned previously, the solubility of oxygen in water is relatively low compared to other gases such as CO2. Let's take a look at this experiment. We have a gas tank, and within this tank, we have oxygen at a partial pressure of 100 millimeters of mercury. So that happens to be the arterial partial pressure of oxygen for humans. So this gas is being pumped into just blood plasma. And so there are molecules of oxygen, which are shown in blue. There are other molecules, presumably something like nitrogen, are shown in another color. So if we look at the actual amount of oxygen per 100 milliliters of this blood plasma, when it's fully saturated, given this partial pressure of 100 millimeters of mercury of oxygen, we only have 0.3 milliliters of oxygen per 100 milliliters of blood. Now let's do the same experiment, but we'll do it with whole blood. Now the red blood cells aren't shown here, but these molecules of hemoglobin, which are shown in red, are presumably inside of red blood cells. They're just not shown here for simplicity. Now we bubble in the same gas, so it has a partial pressure of 100 millimeters of mercury. There's also other gas, such as nitrogen, being pumped into this. And so again, we're going to have the same amount of gas just dissolved in the blood plasma. So again, 0.3 milliliters of oxygen. But if we look at the fact is we have in this case hemoglobin. So the oxygen can bind to hemoglobin, and now we can have a much higher total concentration or oxygen content, which is now going to be 20 milliliters of O2 per milliliter of blood. So we've gone from 0.3 to a much higher total, what's referred to as oxygen content. Oxygen content is defined as the amount of oxygen per unit volume of blood. And so it's much higher. So we've gone up from 0.03 to 20. And so this is basically why we have red blood cells. They're essentially packages for holding hemoglobin. And then the hemoglobin allows us to transport far more oxygen in our blood than we would be able to do otherwise. In the previous slide, we showed that by far the majority of oxygen in the blood is in fact bound to hemoglobin. Let's take a look at the hemoglobin molecule in a little more detail. It consists of 4 polypeptide globins or globular proteins, which you can see here. So there are 2 beta chains and 2 alpha chains. So it has 4 subunits altogether. Each of the subunits has what's called the prosthetic group, which is in this case a heme group. So it has an attached group that's not, it's not a polypeptide. It's a separate type of molecule. And at the very center of each heme group, there's an iron atom. And it's at this iron atom that you can have binding with oxygen. So because there's 4 iron atoms, there are 4 binding sites for oxygen. So one hemoglobin molecule can carry 4 molecules of oxygen. And the red blood cells have so many of these hemoglobin molecules that they're able to carry over 1 billion oxygen molecules. There are different forms of hemoglobin. One form is oxyhemoglobin. This is an oxygen-loaded form of hemoglobin. Another form is deoxyhemoglobin. This is a form of hemoglobin without oxygen. And of course, as we just discussed, there are 4 different oxygen binding sites. So you can basically have different levels of saturation. So if you looked at an individual hemoglobin molecule, it can have 0 molecules of oxygen, so it would be completely deoxyhemoglobin. You could have 4 molecules of oxygen bound to the hemoglobin, therefore you'd be 100% oxyhemoglobin. Of course, you can have intermediate steps, like for example, if you had 2 molecules of oxygen bound to 1 molecule of hemoglobin, you'd be somewhere in between deoxy and oxyhemoglobin. And the point of talking about this is we're going to talk about the saturation of hemoglobin as not just individual molecules, but like say, a lot of hemoglobin molecules within a certain volume. So we'll come back to that idea later. Another type of hemoglobin is carboxyhemoglobin. This is hemoglobin bound with carbon monoxide. So it turns out that carbon monoxide has a very high affinity for hemoglobin. So it's 200 times higher affinity for hemoglobin compared to oxygen. And this presents a problem because you can have carbon monoxide poisoning, which we'll talk about subsequently. Another thing that's important to note here is that each of these different forms of hemoglobin have a unique color and absorption spectrum. So, for example, we can take advantage of the fact that oxyhemoglobin and deoxyhemoglobin absorb light in different wavelengths and different amounts. And so we can use that to determine the degree of oxygen saturation of hemoglobin in a pulse oximeter. As I just mentioned, the hemoglobin has a very high affinity for carbon monoxide, and carbon monoxide binds at the exact same site where oxygen would normally bind. And so carbon monoxide can essentially block oxygen from binding to hemoglobin, and this can have devastating effects because essentially it would mean that the hemoglobin would lose its ability to transport oxygen. Carbon monoxide poisoning may be suspected when the carboxyhemoglobin is greater than 10% in smokers and 3% in non-smokers. Carbon monoxide poisoning is usually caused by the burning of fossil fuels, by faulty appliances, or vehicles within an enclosed environment. So what happens often is someone may set up a generator, and those should always be set up outdoors or in a very well-ventilated space. If you put it in, say, a basement, then this is a deadly combination. It could lead to carbon monoxide poisoning. Symptoms include headache, weakness, dizziness, nausea, confusion, memory loss, psychiatric conditions, and cardiac injury. People may be treated for carbon monoxide poisoning by breathing 100% oxygen at 1 atmosphere, or by breathing 100% oxygen at greater than 1.4 atmospheres of pressure within a hyperbaric chamber. One variable that's often measured to assess a person's cardiorespiratory function is the percent oxyhemoglobin saturation. This is measured as the amount of oxyhemoglobin divided by the total amount of hemoglobin, then multiplied by 100 to yield a percent. It's typically measured using a pulse oximeter or a blood gas machine. And so it's a measure of how well the lungs and the heart are working to oxygenate blood. So the normal value for arterial blood is about 97%, which indicates that for the most part, the hemoglobin are saturated, molecules are saturated with oxygen. There's only a little bit of hemoglobin that isn't saturated with oxygen. The percent oxyhemoglobin saturation value doesn't tell you much about the actual concentration of hemoglobin in the blood, which can actually vary depending on certain disease states. So the oxygen carrying capacity of blood is measured by its hemoglobin concentration. The more hemoglobin, clearly the more oxygen it can carry. However, there are certain disease states, such as anemia, where you have below normal hemoglobin levels. And then there are other disease states, such as polycythemia, which is a high red blood cell count. With this condition, often people have an above normal hemoglobin level. And this may occur naturally when a person moves to high altitude. I should mention that there's a hormone, erythropoietin, which is made in the kidneys, and it stimulates hemoglobin and red blood cell production in red bone marrow when oxygen levels are low. So again, if a person moves to high altitude, there'll be stimulation within the kidney for production of erythropoietin, so it's going to generate more erythropoietin. That will then act on the red bone marrow to increase red blood cell production. The job of hemoglobin is to pick up oxygen within the lungs, within the pulmonary capillaries, and then to take that oxygen and drop it off within the systemic capillaries so that the oxygen can be used by tissue. And the way this works is that the binding of oxygen to hemoglobin is reversible, so it can bind and unbind. And so at high partial pressures of oxygen, such as the partial pressures that are found in the pulmonary capillaries, there's a high saturation of hemoglobin. In other words, it's shown here at 100%. It's probably more realistically about 97%. But for illustration purposes, this is fine. Let's say within the pulmonary capillaries, we have a PO2, or partial pressure of oxygen, of 100 millimeters of mercury. At that level of PO2, we find that the hemoglobin, in this case, it's showing 100% saturated. Now, as the blood moves through the heart, the left side of the heart, through the aorta and the major arteries, there's no diffusion of oxygen at that point yet across the wall. So we would still be at pretty much 100% saturation. By the time we get to the capillaries, systemic capillaries in, say, an organ, then oxygen is being used up, and the capillaries are very allow for the diffusion of oxygen. And so we would get to the point, we would get to start to unload oxygen within those systemic capillaries. And this unloading, of course, occurs because there's diffusion of oxygen away from the systemic capillaries and into the tissue. This reduces the partial pressure of oxygen within the systemic capillaries, and therefore this would then reduce the partial pressure of oxygen within the red blood cells. And again, oxygen is not bound forever. It's bound sort of loosely to the hemoglobin, and so it can unbind. And so you'd have the movement of oxygen down its concentration gradient, its partial pressure gradient, I should say. And so by the time you get to the veins, and this is a person at rest, the partial pressure of oxygen in the veins is 40 millimeters of mercury. So this means that oxygen has moved. It's moved from the blood and into the organs as it passed through, it was perfusing through the organs. And so we can then see that a certain amount of oxygen has been unloaded to the tissue. So an important thing here is just to understand that when you look at this sort of curve, which is on the y-axis, we have the percent oxyhemoglobin saturation. On the x-axis, we have partial pressure of oxygen. At high levels of partial pressure of oxygen, we're going to get essentially a loading of oxygen onto the hemoglobin. Whereas at lower levels of partial pressure of oxygen, we're going to have unloading oxygen from the hemoglobin and into the tissue. The exact numbers aren't really that important, but this slide does give you a little more insight in terms of what's going on. So if we look at the systemic arteries, we have a PO2 of 100 millimeters of mercury. This means that we'll have 97% oxyhemoglobin. Now this corresponds to having 20 milliliters of oxygen per 100 milliliters of blood. For a person at rest, if we measure the systemic blood, we would find that there's a PO2 of 40 millimeters of mercury. This corresponds to a 75% oxyhemoglobin. And this also corresponds to 15.5 milliliters of oxygen. And the point is, you can see that if you look at it this way, that we've gone from 20 milliliters of oxygen per 100 milliliters of blood to 15 milliliters of oxygen per 100 milliliters of blood. Therefore, we have unloaded a certain amount of oxygen to the tissue. And another point I want to make is that, let's say a person is exercising, then the PO2 in the veins would be much lower. Maybe it'd be 30 or 20. And the reason for this is because when skeletal muscle is active, it uses up far more oxygen than it would at rest. And so you'd have essentially a greater extraction of oxygen by these active tissue, and therefore the PO2 would drop even more. And therefore we could look, again, the exact numbers aren't important, but we would go from, say, 20 milliliters of oxygen per 100 milliliters of blood to a much lower number. And that would give us an idea also of how much oxygen is being unloaded to the tissues. This table simply reinforces some of the ideas we've been talking about. With a PO2 of arterial blood of 100 millimeters of mercury, our percent oxyhemoglobin value is 97. So that would be in a healthy person. For a person at rest, the PO2 would be 40 millimeters of mercury, and then you have a lower percent oxyhemoglobin, about 75%. And as I said, the venous blood PO2 could be quite a bit lower. So it could be, way down here, like 23 or something, and that would give you a lower percent oxyhemoglobin value. So at this point, I want to talk a little bit about the actual shape of this curve. So the oxyhemoglobin percent saturation curve is often described as being sigmoidal, because if you look at it, it sort of looks S-shaped. And so a sigmoidal curve starts off sort of with a lower slope, then it has a steep slope, and then it essentially reaches sort of a saturation or plateau. Now, the characteristics of this curve actually have to do with the molecular structure of a hemoglobin molecule. Remember, there are four subunits, four globular subunits. So it turns out for the very first, let's imagine we have deoxyhemoglobin. So there's no oxygen that's bound yet. For the first oxygen molecule to bind, it's a little bit difficult. And so we'd be down here, let's say a low PO2. But once oxygen starts to bind, that after that first oxygen binds to one of the subunits, it makes it easier for the second one to bind, and that makes it even easier for the third one to bind. And this gives rise to this interesting curve where it's very steep. And then eventually, at some point, you're going from, say, the third to the fourth one, and you're going to reach a kind of a saturation point, because remember, the oxygens aren't bound, covalently to the hemoglobin. And so as one binds onto one of the subunits, another one might come off. That sort of thing's going on. And so that's why you get this sort of a curve where it sort of gradually reaches 100% or near 100%. Okay, so this curve actually has some significance, because it means that in the upper range, when we're, say, between 80 and 100 millimeters of mercury, we're going to be very close to 100% saturation. We're going to be around 90, 80, 97, 98, 99, 100. And then when we get into it, and then we get into another point where small changes, where it's very steep here, right? The slope is very steep, where small changes in partial pressure of oxygen will lead to large changes in the percent of oxyhemoglobin saturation, which essentially means that as the blood moves into, say, tissue that has lower PO2, there's a good chance that oxygen will be unloaded. Okay, so what happens under different conditions? Well, we've already discussed rest. So for a person that's at rest, they'll pick up oxygen in the pulmonary capillaries. We'll have an arterial PO2 of about 97%. And then when the blood comes back from the veins, the percent oxygen hemoglobin saturation will be about 75%. And so I think this may seem surprising, because it means that only a small amount of oxygen has been unloaded. So why would you build a system like this? Well, the reason is we're not always at rest. At rest, we really don't need that much oxygen. But once we start exercising, we need to be able to unload a lot more oxygen. And so, only at rest, we only have about a 22% difference in this percent oxygen hemoglobin saturation value. During light exercise, that increases tremendously to 39%. And during heavy exercise, it's actually really, really incredible. But you can actually, this value can go decrease by 80%, which means that we're way down here on this curve, and we'd be unloading a tremendous amount of oxygen. So the oxygen in the veins that's coming back that didn't get used up, that's not really wasteful. It's kind of like an oxygen reserve, And we would only sort of tap into that reserve when we really need it, which is when we're exercising heavily. So far, we've talked about the effect of the partial pressure of oxygen on this percent oxyhemoglobin saturation value, and we've just considered a single curve. In reality, there's actually a family of curves. So for example, if we look at this graph here, we can see on the x-axis, again, we have partial pressure of oxygen, and on the y-axis, we have percent oxyhemoglobin saturation. Now, our standard arterial pressure is pH, is 7.4. What happens when the pH changes? Well, if there's an increase in pH, okay, to 7.6, then this whole curve is said to be shifted to the left. Okay, what does that mean? Well, if it's shifted to the left, that means if we look at a particular partial pressure of oxygen, let's use 40 because that's the value we used for venous before. Here at 7.4, we had 75% saturation, percent oxyhemoglobin saturation. If the pH is higher, if the blood is more alkaline, then we're at, it looks like about 83 or so. So what would this mean? This would mean that there would there's less unloading of oxygen to the tissue if the blood pH is alkaline. So this actually gives you some indication that there may be one of the problems with the pH being too alkaline and outside of its normal range is that it actually interferes with oxygen unloading from blood, from hemoglobin. We've talked previously about the fact that as you change pH, you may change the interactions between various side chains in amino acids and a protein, and this could lead to denaturation of a protein. So that's certainly true, but this is another effect that we're finding here, which is that if the blood is too alkaline, this interferes with unloading of oxygen to tissue. Now let's take it to the other side. Let's say the blood pH is 7.2, so it's more acidic than normal. At this same value of 40 millimeters of mercury, which was our average venous PO2 value, instead of 75% for the percent oxyhemoglobin saturation value, we have 60%. Now, as we've discussed, this would indicate that more oxygen is being unloaded into the tissues. And it turns out that that actually may be a good thing. This whole curve is said to be shifted to the right. You can see it's to the right of this the standard curve, which is 7.4. So why would we want to shift it to the right? This unloading is actually beneficial. So, for example, when a person is exercising, doing aerobic exercise, they're generating a lot of CO2. This CO2 leaves the exercising skeletal muscle, the active skeletal muscle, goes into the blood, and through the reaction that we talked about earlier, an increase in CO2 would lead to an increase in hydrogen ion concentration, which would lead to a lowering of the pH. And this would essentially lead to greater unloading of oxygen into this tissue. So this is actually very convenient because the tissue that's really active would generate enough CO2 and thereby decrease the pH locally within that tissue. And that could actually that does lead to more unloading of oxygen, which is exactly what you want because this is the skeletal muscle that's perhaps more active than other skeletal muscles during a particular exercise. So we're getting really good unloading effects that are local to this specific muscle that's active. So anyway, this overall effect, this affinity decrease at lower pH and an increase at higher pH is referred to as the Bohr effect because he discovered it. And so it turns out that's very important. We get with increased, more unloading occurs at lower pH, as I just discussed. Increased metabolism means more CO2 is produced. This leads to a lower pH within that active skeletal muscle, and this leads to more unloading of oxygen into that active skeletal muscle. And again, this is referred to as a rightward shift of the whole curve. Now, you may notice something, which is with this rightward shift, it affects every point along the whole curve. So if we look at 100% oxygen, which is our typical arterial PO2, we actually have a little bit lower value here. So potentially this might have a little bit of an effect on the loading of oxygen within the pulmonary capillaries if the blood continues to be a little bit more acidic by the time you get to the pulmonary capillaries. However, because of the nature of the shape of this curve, this difference is really fairly insignificant. Whether you're at 95% saturation versus 97% really doesn't make that much difference. You're going to load up a lot of oxygen. And the more significant thing is how well can you unload that oxygen within the active tissue. So again, that's why this Bohr effect is incredibly important for active muscle. This table simply gives us more information about the actual amount of oxygen that would be unloaded. So let's take our normal pH of blood, 7.4. Here's our arterial O2 content, 19.8. Here's our venous O2 content, and therefore the amount of O2 unloaded per tissue to tissues per 100 milliliters of blood is 5.0 milliliters of oxygen. Let's imagine that the pH is lower, 7.2. This means there's going to be a decrease in affinity, which we can see here as the right shift in the curve. The arterial O2 content is a little bit lower, which is what you'd expect because of this decreased affinity. So it's now 19.2. But then if we look at the venous O2 content per 100 milliliters of blood, it's only 12.6. And if you just do this difference, you find that more oxygen has been unloaded to tissue per 100 milliliters of blood. So we have 6.6 milliliters of oxygen unloaded versus 5.0. In addition to pH, temperature also has a large effect on hemoglobin affinity for oxygen. At higher temperatures, oxygen has a lower affinity for hemoglobin, so that there's a decreased binding. This actually further enhances the amount of oxygen that's unloaded to muscles during exercise. So for example, the whole curve is shifted to the right, just as the whole curve was shifted to the right with the lower pH. Now this means that for a given partial pressure of oxygen value, there'll be a lower percent oxyhemoglobin saturation value. And we've already established that that means there's going to be more unloading of oxygen to the tissues. And so exercising muscle, of course, is warmer than non-exercising muscles because of the friction within the muscle, and also due to the fact that all the cellular processes are not 100% efficient. So this also helps to bring oxygen specifically to the muscles that are the most active. There's yet another factor that affects the percent oxyhemoglobin saturation curve. And this factor is 2,3-DPG. Red blood cells obtain energy from anaerobic metabolism of glucose. And this is because red blood cells have no nucleus and no mitochondria. During this process, 2,3-diphosphoglyceric acid, or 2,3-DPG, is made. And actually, it turns out that in this process, the enzyme that is involved in the production of 2,3-DPG is inhibited by oxyhemoglobin. So this means when there's a high percentage of oxyhemoglobin within a red blood cell, this production is inhibited. However, if a person is at high altitude or if they're anemic, then there'll be a lower percentage of oxyhemoglobin within the red blood cell, and therefore this enzyme that leads to the production of 2,3-DPG is not inhibited, and therefore there's more production of 2,3-DPG. So 2,3-DPG has a very similar effect to the effects we talked about with temperature and with pH. So an increase in 2,3-DPG within the red blood cell leads to an increase in oxygen unloading. So this means at a given, say, partial pressure of oxygen. So this means that the whole oxyhemoglobin dissociation curve is shifted to the right, just as it was with higher temperature and just as it was with lower pH. And this is actually helpful because if a person is anemic, that means they don't have enough hemoglobin in their blood. And a way to make up for that is to unload more oxygen within the tissues. Similarly, for a person at high altitude, unloading more oxygen to tissue is a good thing, and so that helps the person who's at a high altitude. This table summarizes the various effects we've talked about on the oxyhemoglobin saturation curve. Notice that the term dissociation curve is used here. That's simply another way of describing the same curve. First of all, a decrease in pH leads to a decreased affinity between hemoglobin and oxygen. This leads to a rightward shift of the entire curve. This is called the Bohr Effect, and it helps in some sense because it increases oxygen delivering during hypercapnia. So hypercapnia is an increase in CO2 production, and this would occur when, for example, a person's exercising and they generate a lot of CO2 through aerobic metabolism, which of course leads to an increase in hydrogen ion concentration and a decrease in pH. Similarly, an increase in temperature decreases the affinity between oxygen and hemoglobin. This also shifts the whole curve to the right. This increases oxygen unloading during exercise and also during fever. An increase in 2,3-DPG also decreases the affinity between oxygen and hemoglobin. Again, it's going to shift the whole curve to the right. This increases oxygen unloading where there's a decrease in total hemoglobin, such as in a person who has anemia, or in a person in which there's a decrease in total oxygen content, which should occur at high altitude. And so this shift to the right means that there's improved unloading of oxygen to tissue. And so this is an adaptation that helps a person with anemia, and it also helps a person living at high altitude. There's yet another respiratory pigment, and this is called myoglobin. This is found in skeletal and cardiac muscles in the human. It can actually be found in smooth muscle in other animals, such as, for example, if you look in a chicken, there's a gizzard which grinds food. That's smooth muscle, and yet it also has myoglobin. But in human, as far as I know, it's restricted to skeletal and cardiac muscle. So this molecule is a little bit different than hemoglobin. Notice that instead of having 4 subunits, it's just one globular protein. It's similar in the sense that it has a prosthetic group, which is heme, and it will bind oxygen. So instead of having 4 oxygen binding sites, it has a single oxygen binding site. And because of that, this difference in the molecular structure actually has an impact on its curve. It has a much simpler curve than hemoglobin. So here we have the percent oxygen saturation versus PO2, and you can see that it's kind of a very simple curve compared to the hemoglobin curve, which is sigmoidal. This is more just a curve that increases and then sort of saturates. You'll also notice that myoglobin has a higher affinity to oxygen compared to hemoglobin. And there's actually a reason for this. So functionally, this myoglobin sits within muscle cells, and it serves as a kind of, it says here, go-between in transferring oxygen from blood to the mitochondria. It also serves as a kind of temporary store or reserve of oxygen. So what happens is oxygen moves down its partial pressure gradient from within a systemic capillary into interstitial fluid and then into cells, such as muscle cells. Then within the muscle cells, you have the mitochondria that are actively doing aerobic metabolism. So oxygen can then move down its partial pressure gradient into the mitochondria. So this could all occur without myoglobin, but myoglobin has a higher affinity for oxygen than hemoglobin. So it's almost like a sort of a waypoint, like the oxygen can then bind to myoglobin, at least temporarily. And then due to the fact that the mitochondria are reducing the partial pressure of oxygen so much, the oxygen is not bound very tightly to the myoglobin, so then it comes off, and then it will move into the mitochondria and be used up and essentially converted to water. So why have myoglobin? Well, it serves as a sort of temporary buffer. It keeps the oxygen flowing essentially to the mitochondria, even though there might be some situations in which the blood flow is not perfectly constant. A good example of that would be cardiac muscle. During systole, because of the constriction on the heart, there's really very little blood flow through the coronary arteries. It's just too much stress on the heart. So the coronary blood vessels get filled with blood during diastole, actually, mostly. And so essentially with each heartbeat, there's a period of time where there's hardly any oxygen coming in directly from the blood. And so the myoglobin is a perfect storage vehicle. So it would hold the oxygen temporarily, give it up to the mitochondria, and then that would be during systole, and then during diastole, it would get replenished. The oxygen would get replenished onto the myoglobin. So it serves this kind of temporary reserve function. And also another argument for the fact that this is what myoglobin's purpose is if you look at diving animals like whales and seals and so forth, you'll see that their skeletal muscle that's involved in swimming is extremely dark to the point of being almost black. And the reason for that is due to myoglobin. So they're just so filled with myoglobin. So again, the myoglobin there is serving as a real reserve of oxygen for the skeletal muscle when the animal is diving underwater. We'll look next at carbon dioxide transport. Carbon dioxide is carried in the blood in three forms. First of all, it's dissolved in the plasma. CO2 is more soluble in water than oxygen. It's about 20 times more soluble. So about 10% of the CO2 that's transported is transported in this form, dissolved. It's also transported as carbaminohemoglobin. That is, it enters into a red blood cell and it combines with hemoglobin by binding to an amino acid. And this accounts for 20% of the CO2 that's transported in the blood. Finally, the last portion, or 70% of CO2, is transported as bicarbonate ions. As I just stated, carbon dioxide is mostly transported in the form of bicarbonate. How does it turn into bicarbonate? Well, within each red blood cell, there's an enzyme called carbonic anhydrase. And this catalyzes the reaction between water and CO2 to form carbonic acid, which is shown here. Water and CO2, when they collide with the aid of carbonic anhydrase, they can form carbonic acid. Now, they would naturally do this reaction without the enzyme, but the enzyme greatly speeds up the reaction rate. And so basically what's happening is that this is going on within a red blood cell. CO2 and water with the aid of carbonic anhydrase leads to the formation of carbonic acid. Within a red blood cell, the carbonic acid that's been formed will dissociate into bicarbonate and hydrogen ions. Due to the action of the enzyme carbonic anhydrase, we now have the formation of hydrogen ion, which is a positive charge, and a bicarbonate ion, which has a negative charge. The hydrogen ion can combine with hemoglobin, and it's binding it to a site other than where the oxygen would bind. Now we have a little bit of an imbalance. We have bicarbonate, and it can now move down its concentration gradient out of the cell. As it's being formed due to the breakup of carbonic acid into hydrogen ion and bicarbonate. As it moves out, however, this constitutes the movement of a negative charge from inside of the cell to outside. So this would mean that the inside of the cell would become positive with respect to the outside. And as this happens, chloride, which is negative and is found in outside of the cell, it will move into the cell. So it's attracted to the positivity inside of the cell, and bicarbonate is moving down its concentration gradient from inside to outside of the cell. This exchange of these two negative ions is referred to as the chloride shift. The bonding of the hydrogen ion to the hemoglobin lowers the affinity of the hemoglobin for oxygen, and this helps with unloading. In fact, this is the Bohr effect, which we spoke of earlier. And interestingly, the unloading of oxygen actually increases from hemoglobin, increase the affinity of hemoglobin for hydrogen ion. And this allows for even more hydrogen ion to bind to the hemoglobin, which helps the blood carry more carbon dioxide. In the pulmonary capillaries, a lot of the things we just talked about are all simply reversed. So there's an increase in the partial pressure of oxygen because, of course, there's a high partial pressure of oxygen within the alveolar airspace. This favors the production of oxyhemoglobin from deoxyhemoglobin. As deoxyhemoglobin is converted to oxyhemoglobin, this leads to a lowering of the affinity between hydrogen ion and hemoglobin, so the hydrogen ion dissociates. So now we have excess hydrogen ion. It can recombine with bicarbonate that happens to be within the red blood cell, and this will then form carbonic acid. As this happens, this reaction goes forward, there's a reduction in the amount of bicarbonate. And so now bicarbonate will diffuse back into the cell. And as it does that, that's negative charges moving into the cell. So this repulses chloride, so it will diffuse back out of the cell. So we have essentially a reverse chloride shift. So now we're producing carbonic acid. And in this low PCO2 environment, so the partial pressure of CO2 is lower in the alveolar airspace compared to the pulmonary capillary. So this means that there's a net movement of CO2 down its partial pressure gradient from the blood plasma and into the alveolar airspace. So under those conditions, carbonic anhydrase will actually convert carbonic acid back into CO2 and water. And the CO2, of course, is exhaled. And so this keeps the reaction going in this direction where we're essentially pulling more and more CO2 out of the form it had been in, which is bicarbonate.